Ирина Владимировна, здравствуйте. Я почему-то вас не слышу. А вы меня слышите?
Сейчас я включу.
Вот, я теперь слышу.
Угу. Ой, не могу установить виртуальную заставку. Пишет,
Ничего
что
страшного.
поддерживает.
Бывает такое, у нас есть такие, да, участники. Ничего страшного.
Ничего страшного. Ну хорошо, тогда как бы что-то еще или ждем просто?
Вы меня слышите? Ждем всех тогда.
Ждем. Ой, не могу установить виртуальную заставку.
Ничего страшного. Бывает такое, у нас есть. Да.
Хорошо, тогда как бы что-то еще или
Ждем всех тогда.
ждем?
Hello, everybody. Can you hear me?
Hello, yeah.
I think that
Hello, yes.
we could start because it's already 3 p.m. and we have our first participant. Firstly, I would like to say hello, all of you. To say thank you to all of you, to participants, to your supervisors, and we are really glad to see you at our international forum. Let's start. Our first team from Indonesia, Neiza Farah, Najmasari Muzena Hisaim Opah. Okay, guys, you can start.
Okay, may I share my screen right now? Hello?
Hello, hello.
Hello.
Okay.
Hello.
All right. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, I'm Neiza Farah Najmasari. I'm here with my two partners,
Muzena Hisham Uqbah,
and Amalia Syahbani Zulfina Putri. And we are here to present our study in female healthy mask. I'm sorry. All right. We are here to present our study in female healthy mask, utilization of Sansevieria and Melaleuca extracts as a common acid absorbent and antibacterial mask. So, firstly, protecting the body's immunity during this pandemic is a very important thing to do. One of so many ways we can do is to keep away from bacterial and any dangerous gas that can cause a drop of the body immunity. Therefore, female healthy mask, which is a mask with filter that contains pure extracts of Sansevieria and Melaleuca, could be the innovation for a carbon monoxide absorbent and antibacterial mask. There are four methodologies used in their research: maceration, carbon monoxide absorbent test, disk diffusion method, and phytochemical test. The phytochemical test is an additional test we did, so the result is not available on the full text.
So, the methodology is maceration. Based on Jamie Jumadi's research in 2018, the maceration method is used to get Sansevieria. Some tools and ingredients that are needed for the maceration are digital scale, Baker glass blender, plastic cone, glass jar, filter paper, rotary evaporator, Sansevieria and alcohol 96%. The procedure starts with preparation, and the first step is cutting the Sansevieria leaves into small pieces and dry it in the sun. Second, blend the dry Sansevieria and weight of dry Sansevieria and alcohol 96% with the 1 and 5 comparison. Then mix them well in the glass jar and put in the dark place. Leave it for 72 hours long. When the 72 hours has already done, filter the extract using filter paper and evaporate in the rotary evaporator. The last step is dip the filter paper to the fusion. Based on Susanto from Padian UPTK 2020, some tools and ingredients that are needed for the carbon monoxide test are gas analyzer, which have we used ECOMD, hoses, motorcycle, filter paper with Sansevieria extract, and plastic bottle. The method will start with cutting the plastic bottle in half and put the filter with Sansevieria extract between the bottle, cuts the attached with tip. The next step is making two holes at the top and the bottom of the bottle. Then put two different holes to close holes. After the bottle are already set up, turn on the gas analyzer and make sure the carbon monoxide degree is 0. Then put the hose at the bottom of the bottle to the motorcycle exhaust and the hose at the top of the bottle to the exhaust probe. Then turn on the motorcycle start to observation and focus to the carbon monoxide degree on the gas analyzer screen.
The next one is disk diffusion method. Based on Valery Sadikian ibn Sauda in 2016, the disk diffusion method is held to know the Klebsiella pneumoniae antibacterial properties of Melaleuca. Agar plates are inoculated with a standardized inoculum of the test microorganism. Then, filter paper discs as wide as 6 mm containing the test compound at the desired concentration are placed on the agar surface. The petri dishes are incubated under suitable conditions. Generally, antimicrobial agent diffuses into the agar and inhibits determination and growth of the test microorganism, and then the diameters of inhibition growth zones are measured. This method is held by Saraswati Indo-Kanatage Committee, and we receive only the result of the test. The next one is phytochemical test. For the identification of phenol, 1 ml extract of Sansevieria from the ethanol solvent is inserted to a test tube. Following that, methanol is added to the tube. Shake well until the solvent turns homogeneous. For testing, we add ferric chloride, then we observe the solution and conclude the result.
I'm sorry. For indication of flavonoid, we use 1 mm extract of Sansevieria was inserted of the test tube. Then add 5 drops of ethanol and then mix it up more homogeneous. Then add magnesium reborn and 5 drops of hydrolytic acid.
And for the identification of tannin, 1 ml of Sansevieria extract is added to the test tube. We use ferric chloride with 30% molarity as the reagent for this experiment. We added some of the reagent to the same test tube, and lastly, we mix the solution until it's homogeneous. After that, we will observe the solution and conclude the results. The last one is alkaloids. Alkaloid identification is done by Mayer, Wagner, and Degendorf methods. Firstly, pour 0.5 grams of Sansevieria extracts into a test tube and add 1 ml of hydrochloric acid and 9 ml of aqueous. Heat them up for 2 minutes, and after 2 minutes, divide them into 3 tubes and leave them to cooling down. After the temperature is normal, add the Mayer, Wagner, and Degendorf solvent into each tube and mix them well and take a conclusion about the result. And here are some results and analyses of carbon monoxide adsorbent test, disk diffusion method, and phytochemical test.
Carbon monoxide adsorbent. Based on carbon monoxide adsorbent test, it indicates that reliable filter that contains Sansevieria and Melaleuca extract has the best result of the carbon monoxide screening.
Based on the disk diffusion method, it indicates that Melaleuca extracts has a clear zone diameters in the amount of 7.92 mm. According to Jeffs and Scout in 1997, this diameters of clear zone is classified into a medium level of Klebsiella pneumoniae antibacterial. And the next one is phytochemical test. The result for alkaloids. Based on Herber research in 1987, reaction from Mayer retraction is form white sediment, while the Degendorf retractions form pink-orange color, and the Wagner retractions form a dark brown sediment. And here we can see that Mayer method form a clear yellow color with yellow sediment, Wagner method form a brown color with black sediment, and the Degendorf method form a pink-orange color, which means the Sansevieria extracts has an alkaloid content from all used methods.
Based on Paulinus 25, the positive rejection in the yellow changes. Here we can see that it's yellow, meaning Sansevieria extract has a flavonoid content. Based on Satyavati on 2014, the result of the panel identification shows that the solution turned into yellow. It means the solution content panel. And then in the identification of tannin experiment, the solution turned into dark green. That also means that it contains tannin.
And here is the mask design of our product and some documentations of our research. So the conclusion from our studying is that filter paper with Sansevieria amelica extracts have the potential to become an acetaminophen and antibacterial mask. Meanwhile, further research is needed to know the expired time of the filter paper with Sansevieria amelica extracts. And Charlie Gilkey once said, "If it matters to you, you'll find a way." And this is matters to us, and we found a way to be here today. And thank you so much for your attention. Goodbye.
Thank you very much. It was a very interesting project. Can I ask you some questions for it? At first, is your product more effective than analogs? Thank you.
So, hang on a second. Compared to other medical masks, it's actually the same because they still can be antibacterial mask, but here the special thing from our mask is that it has the ability to improving the airway. So it has double function. The first is antibacterial and the second one is ability to improving the airway. Thank you.
Thank you. And please, one more question. Do you think it is possible to obtain such material on an industrial scale?
Pardon me, can you repeat the question?
Do you think it is possible to obtain such material that you do on an industrial scale? Don't understand?
Sorry.
Oh, can you expand your production for industrial scale? Do you understand this?
Oh, Dr. Scale?
Is it enough Sansevieria to produce this mask in much more
Industrial volumes. Industrial
yes.
volumes, yes.
So this mask is, the Sansevieria is like very common plants in Indonesia, so we can find in every corner of the country. So maybe it is possible to produce more of the mask filter.
Thank you.
Could I ask a question about toxicity? What can you say about the toxicity of your extracts for people?
And there's also some further research that needed because we haven't done the toxicity test yet.
I have one question. Can you clearly state the purpose of your study, please, once again?
The purpose of this studying is to make a filter, I'm sorry, a mask with filter that contains Sansevieria in Melaleuca. And the Sansevieria filter is there to filter carbon monoxide gas, and the Melaleuca filter is there to be an antibacterial stuff.
Okay, and do you think that the components of this mask are pure enough to be used in such a medical use?
For the Sansevieria, it is pure extracts because we do the extracts ourselves. But for the Melaleuca, we did not know how the extracts was extract because we just received a result for it.
Okay, thank you.
You're welcome.
Thank you very much for your presentation. Let's move to the second team, the team from Indonesia. Are you ready? Please introduce Alisa and Carmeliza Alder Rustam. I can't hear you.
Can you hear us?
Yes, that's great. Yes. You can start.
So, hello everyone. Greeting, thank you so much for your opportunity. So I am Armeliza Rustam.
I'm Quinta Feris Kalisa.
So we are going to do a presentation about our research. And our research is about CFC, chlorophyll fuel cell. Electricity test on chlorophyll of papaya leaves as a green power source using a chlorophyll fuel cell system or CFC system. So research has been made about the source of electricity produced from the chlorophyll of papaya leaves or Carica papaya L. And it has not been made much use of chlorophyll as a produce of electricity, since power production is dominated by the use of fossil fuels. In the research, the technique is to extract papaya leaves by using ethanol 70%. Cut and pounded, the papaya leaf is then soaked in ethanol for 1 day. And the extract of papaya leaves is used as a cathode by using the voltage cell technique to determine the electricity generated.
The background of the study itself, it should be noted that the efforts to find electricity sources are constantly being made, and with the invention of these new alternative energy sources, they may replace this depleted little by little, while alternative energy sources may not be as optimal as mineral energy sources, but continuing to the research can optimize alternative energy to be more effective, cheaper, or affordable and environmentally friendly.
So one way to produce alternative energy sources is to make use of the chlorophyll in papaya leaves, which because papaya leaves have been known to have the highest chlorophyll content among plant leaves that can be used as food supplements such as spinach leaves and others. Additionally, selecting papaya leaves as a source of chlorophyll for electricity sources is because papaya trees are readily available and are very environmentally friendly. Also, the purpose is to obtain voltage values and other electrical properties from the designed AHA extract using a chlorophyll fuel cell system. Here is the material that we will need to do this experiment: a papaya leaf, an alcohol 90%, an aluminum foil, a dual chamber CFC system, a multimeter tester, and a pair of scales. Step 1. Here's the sample preparation. And also you need an extract of papaya leaf. The papaya leaf small cut and then smoothed afterward. Step 2. The extract is fed into a glass and left for 24 hours with aluminum foil to prevent evaporation. Next, the extract of papaya leaves soaked with screened ethanol used sari paper to separate the extract from the unextracted leaves. Step 3. Preparation of the power source. After producing chlorophyll extract from papaya leaves, the next step is to create a system to test the electricity produced. The system was created using photocell principles with chlorophyll extract solution as a cathode water and water as a cathode. And the last step is to analyze. The photocell system runs for 20 minutes to see the performance of the system and most electricity generated. The resulting data sampling of electricity voltage is observed every 2 minutes using a digital multimeter.
The result and a short explanation in the research are to test the electric voltage generated in the experiment. Two variations of the electrode were used, namely carbons and CuZn. The use of carbons electrodes in order to produce the pure voltage generated by chlorophyll of papaya leaves, because carbon electrodes are inert electrodes that are not easy to react to. And so, to get the optimal voltage, CuZn electrodes are used. So here's the result of voltage versus time graph of using CuZn electrodes makes the electric voltage increase. Apparently, that any increase in the mass of papaya leaves would result in an increased electrical charge. This is seen in the mass of 12 grams of papaya leaves. The initial voltage produced is 937 millivolt in the state of electrodes is carbons, while the smallest value in the experiment produced is 6088 millivolt. The discussion and conclusion in this research are that it appears that each additional mass of papaya leaves, the greater the voltage generated. From all the voltage results obtained, the average voltage generated using 12 grams of papaya leaves and the carbon electrode is 4731 millivolt. The average voltage generated using 24 grams of papaya leaves and a carbon electrode is 177.78 millivolt. And lastly, the average voltage generated using 48 grams of papaya leaves and a carbon electrode is 152.2 millivolt. The conclusion of the experiment that this is proven and obtained through the voltaic cell theory and the redox reaction. From experiments carried out using 3 subjects and 2 different electrode pairs, carbons and CuZn, it was found that papaya leaf extract can produce between 37.1 to 937 millivolt of electrical energy. The average voltage generated is 434.28 millivolt. The things that affect the size of the voltage are the mass of papaya leaf itself and the type of electrode pairs used. And that's all. Thank you so much for listening to our research. Have a great day.
So I have one question. First of all, thank you for your demonstration. And I just wanted to ask, maybe I just didn't quite catch this moment, but why did you choose papaya leaves in particular?
Because it contained the most chlorophyll content among other plant leaves. So the average level of chlorophyll leaf is found at the top of the leaf is 2.76 milligrams per grams, and the lowest level of chlorophyll is found at the 23rd with 1.59 milligrams per gram. So that's why we use papaya leaves.
Okay, thank you.
Hello, thank you for your presentation. I have a few questions. First of all, what is the lifetime of your batteries?
So it actually depends by the sun.
Can you say approximately how much can it be powerful? For how long can it be powerful?
We haven't counted the power density yet.
Okay, thank you. And how how much papaya do you have to build this battery?
Are you asking about how many papaya leaves that we used in the experiment?
Yes.
Only a papaya,
One papaya
one papaya leaf.
Okay, thank you.
I have one question, if I may. Did you modify chlorophyll from papaya leaves? I heard that it is unstable in direct sunlight in long term.
Um, um, So that's why we use the CFC system, because to find the pure voltage from the chlorophyll papaya leaves and to optimize the voltage itself of using CUZN, so it can increase. I hope that helps.
Okay, thank you.
I have one more question. May I? Did you compare your batteries with other ones that are similar to yours?
No, we haven't compared yet.
And why didn't you?
Because because so we're still developing our experiment, our project.
Okay, so this is just the beginning of your research. Okay, thank you very much.
Thank you so much.
Thank you so much.
Thank you very much for your project. Can I ask one question? Why did you choose papaya leaves for your research?
Because it contains the most chlorophyll content among other plant leaves. Yeah, that's why we use papaya leaves.
And how do you see the continuation of this project?
Continuation?
Hello.
Sorry, can you repeat the question once again? Sorry.
How do you see the continuation of this project? Extension, continuation, prolongation.
We're still developing some
project.
Yeah, in the system itself. So it's on yeah, it's on a development thing.
It will be developed into a biobattery.
Sorry? Can you repeat?
It will be developed into a biobattery.
Oh, okay. Thank you.
Thank you very much. Let's move to questions. If not, we shall continue. Thank you very much, Lisa and Rita. Next, we shall welcome the team from Indonesia.
I'm sorry, but we can't hear anything.
You can hear us. You are welcome our next team.
I'm sorry, Mr. We cannot hear anything.
Can you hear me? Hello?
Your voice is too low.
Меня слышно? Нет, меня слышно?
Yes, now we can hear you.
Лучше, да?
Немного прерывисто слышно.
На самом деле очень тихо и почти ничего не понятно.
Сейчас разберемся, какие технические неполадки небольшие. Okay, do you hear me now?
Yes, sir.
Yeah, next team from Indonesia, Muftafik and the partner, please start your presentation.
The honorable jury, the organizer, participant, and audiences in this meeting room. I'm Mulfianingsi with my partner Muhammad Taufik M from Alauddin State Islamic University of Makassar, Sulawesi Island in Indonesia. I would like to say thank you very much for this section. It is a great opportunity for us to present our research in this conference, and the title of our research is QSAR analysis and design of a new amino alkanol xanthone derivative compound as anti-cancer. For introduction, cancer as one of the diseases that can cause relatively high mortality in the world. Cancer is the generation that is widely feared by the public because the number of patients continues to increase. It's caused by cancer cells. Cancer cells have tissue cells are abnormal and tissue growth is quickly. One of the compounds that serve as a deterrent to cancer is xanthone compounds, and xanthone compound can be found in mangosteen fruit peel. This is evidenced by the results of the study that mangosteen fruit peel contains flavonoid, tannins, and xanthone that have pharmacological effect. Xanthone derivative compound can be designed through computational chemical modeling. It can cause computational chemistry as one of the chemical that can design of medicinal compound. with quantitative relation between structure and activity using Austin Model 1 method. Next, the purposes of our research are: the first, to find out descriptor that can influence the anti-cancer activity of xanthan amino alkyl derivative compounds, and the second, to know the best quantitative relation between structure and activity, or HKSA equation model in xanthan amino alkyl derivative compound against anti-cancer activity using multilinear regression analysis. And the last, to know the predicted anti-cancer activity results of new compounds, modified amino alkyl xanthan derivative compounds. And ladies and gentlemen, this is the basic structure of xanthan compound. Next, there are 15 compound name and value of xanthan amino alkyl derivative compound activity from Skaradex et al. Research in 2016. And based on the table, we can see that each structure that has a different activity value or IC50 value. Next, this is the previous research before our research about xanthan derivative compound. But in this research, just Arba et al. Research at 2016 that have predictive compound. Research method in this research has been done in December 2020 until January 2021 at Organic Chemistry Laboratory, Faculty of Science and Technology, Alauddin State Islamic University of Makassar. And next, for hardware for device on substance in our research. The first, we have two type of hard device. That is, there are hardware and software, and we can see the hardware and the software for our research. And for substance, we use amino alkyl xanthan derivative compound from Skaradex Research at 2016. Next, this is the procedure of our research. Actually, we have three main procedure. The first, geometry optimization, statistical analysis of HKSA, and the last, calculating value of predictive anti-cancer activity. And for the research line, we can see in this slide, we have to start, and we get secondary data from Skaradex Research at 2016, and we make two sets, the first training sets and the second test sets. For training sets, we have to selection and calculation of descriptor, and after that, building up of HKSA equation. And for test sets, we have to selection and calculation of descriptor, and after that, validation of HKSA equation. And after that, we got HKSA equation, and after that, we have to prediction of anti-cancer activity of amino alkyl xanthan derivative compound, and the research is finished. For geometry optimization, we can see in this slide, this is the procedure of geometry optimization, and this is the procedure of statistical analysis of HKSA, and this is the procedure of calculating value of predictive anti-cancer activity. This presentation will be continued with my partner.
Okay, next result and discussion. The decision of new compound is done by determining the parent compound and selecting the position of the side chain in which the carbon atomic position is selected in the study.
Sorry, we can't hear you. Can you turn on the microphone, please? We can't hear you. Can you turn the microphone on, please? Thank you.
Okay. The design of new compounds is done by determining the parent compound and selecting the position of the side chain in which the carbon atomic position is selected in the study. The selected substituent should be electronegative in order to increase the electronegativity value of the new derivative compound. Furthermore, the design of the new compounds was carried out using the best QSAR equation formula. Then the geometry optimization and descriptor calculation were carried out again until the predicted log IC50 value was obtained. Next. The data structure. The data structure is divided into two data sets, namely training sets and test sets. Descriptor must represent hydrophobic, electronic, and steric parameters connecting of 14 descriptors, namely HOMO energy, LUMO energy, atomic networks, hydration energy, polarizability, total energy, dipole moment, surface area, molecular mass, molar refractivity, volume, height of formation. The following shows the value of each descriptor. In the Kishore equation model, next. There are 5 models with different values. In each model, there are variables that are excluded, which means that these variables are eliminated until the best equation is obtained by considering the assessment criteria in the form R squared, F count, F table, F count per F table. The best equation model is in model 3 with a value log IC50 4.308, total energy negative 0.0004, surface area negative 0.014, dipole moment positive 0.174, height of formation positive 0.0002, carbon 1 positive 14.2114, carbon 2 positive 1.137, carbon 3 negative 13.557, and carbon 10 23.059. Which prove that the descriptor combination is an influent physicochemical property. From the research that has been done, it has obtained the atomic networks, total energy, and dipole moment, which represent the electronic parameters, surface area, and height of formation, which represent steric parameters. In model 3, shows the active center of the xanthan amino alkyl derivative compound is located the networks of atom carbon 3, carbon 10, carbon 2, and carbon 1. The design of new compounds is done by determining the parent compound and selecting the position of the side chain in which the carbon 3 atomic position is selected in the study. The selected substituent should be electronegative in order to increase the electronegativity value of the new derivative compound. Furthermore, the design of the new compound was carried out using the Best-Kussar equation formula. The geometry optimization and descriptor calculation were carried out again until the predicted log IC50 value was obtained. Conclusion: descriptor that can affect the anti-cancer activity of xanthan amino alkyl derivative compounds such as clean charts of atom total energy, tissue surface area, head formation, and dipole moment. The equation model of structure activity quantitative relationship selected in xanthan amino alkyl derivative compound against anti-cancer is model 3. Obtain the results of prediction of anti-cancer activity from new compounds modified amino alkyl xanthan derivative compound that more potentially inhibit the activity of cancer cell. Our modified compound is 3,2-hydroxy 3-propan-2-il-amino propoxy 9-xanthin 9-on and 3-chloro 6,2-hydroxy 3,2-hydroxyethyl methyl amino propoxy 9-xanthin 9 on with a log IC50 value of 1.84 and 1.59. Thank you.
Thank you for your presentation. Does anyone have any questions?
What kind of software do you use for modeling your compounds?
For chemical modeling,
Okay, thank you for the question. For chemical modeling, we use software is CHEMS Hyperchem for Hyperchem type 8.0.7.
Have you any experience before this experiment? Have you any other models before this experiment? Have you any experience before, earlier?
Thank you for the question. Before our experiment, we have data from Skaradek research in 2016, and the Skaradek research that have 15 derivative compound of
Hello, can you hear us? It seems they have some kind of technical difficulties.
Okay.
If they want to increase next minute, we'll have to move to the next team.
Sorry, I couldn't understand you.
If the current team won't unfreeze in the next minute, we will have to move to the next team.
Okay. So what should I do now? Sorry.
I'm sorry, sir. We have a trouble connection.
Your microphone is muted.
Yeah, no, it's not.
Okay, sir. Can I answer before question? Question before this. Okay. For the answer for the question. Before we are experiment this, and we have data from Skaradek research at 2016, and we can see in the Skaradek research 2016 have 15 compound aminolcanol derivative compound, and we use that for our research, and we make new design from that research. Maybe just that.
And determine the best question model by considering several assessment criteria and press scores. Thank you.
Thank you very much. Thank you for your presentation. Let's move next to the other team from Luxembourg.
Hello, can we share my screen to show the presentation?
Yes, one moment. Can you demonstrate your screen?
Yeah. So can we start with our presentation? Okay, I'm just gonna start the presentation. So good afternoon, ladies and gentlemen. I'd like to introduce myself. My name is Jennifer Michael, and my teammates are here. Anna Batsmanova. Hello. And Kajal Karki. Mayan. And today we're gonna explain to you our presentation, which is the degradation of plastic polymers using genetically modified bacteria. So why did we choose this project? We wanted to find a sustainable method to reduce plastic pollution in Luxembourgish rivers and rivers worldwide. Plastic pollution is known as the accumulation of synthetic polymers in the environment, where they cause problems in wildlife and their habitats, as well as for human populations.
Plastics are non-biodegradable, meaning it'll take them hundreds and thousands of years to be broken down in nature, which then causes an accumulation of plastics in wastelands. So with the project, we want to find a sustainable method to speed up this process of degradation and so reduce plastic pollution.
The another reason for doing this project is that during my internship week at University of Luxembourg in biology department, I saw how much plastic was used every day. After that, I discussed this with my friends about the increasing use of plastics in our daily lives, which encouraged all of us to find a solution of this increasing plastic crisis. And after doing some research, we then come up with a few interesting ideas.
So this is a proposed method. Unfortunately, it is all only theoretical. We couldn't carry out this experiment due to COVID-19 restrictions.
The first step of our method was to insert a foreign DNA into our vector Escherichia coli, also known as E. coli. Vector is a means of delivering genes into a cell using gene technology. The reason for using E. coli is that firstly, it is easy to access. Secondly, it is cheap, and most of the strains of E. coli are humans. Then Escherichia coli transcribes the gene on foreign DNA that codes for the enzyme.
The bacteria then translates this code to produce the amino acids, and then multiple amino acids come together to form our enzymes.
We then tested how these enzymes break down plastic, specifically PET.
The summary of our method. So we assumed that we would start off with only one strand of DNA and E. coli bacteria. The method we use to multiply our DNA to a big enough sample is called polymerase chain reaction, also known as PCR. This is a type of technique commonly used in forensic sciences. To modify the bacteria, we use the restriction enzyme to cut the part of plasmid of E. coli. And DNA ligase is used to link together sugar phosphate backbone of the DNA molecule, coding for protease and metase and the plasmid, which produces a closed circle of double-stranded DNA.
The next step is to insert the modified plasmid into the E. coli. This is done by adding concentrated calcium ion solution to the bacterial cultures. The bacterial cultures are then given a heat shock to make sure that the plasmid passes to the cell surface membrane. The next step is the identification of the modified E. coli. The initial DNA coding for protease and metase is given a genetic marker, specifically GFP extracted from jellyfish. GFP fluorescence when exposed to UV light. So our expectation is that the E. coli bacteria will fluorescence when it's exposed to UV light, which tells us that it has been taken up by the modified plasmid. Hence, the uptake of the bacteria by modified plasmid is proportional to the intensity of fluorescence when exposed to UV.
Once UV light was shown onto the bacteria, some of them would have been luminescent because if the gene was taken up, then the luminescent protein could have been produced, which is what we detected. The luminescent marker was used instead of an antibiotic resistance marker because the results were easier to visualize and the whole process would have been less cumbersome. So then the next step would have been to clone the bacteria that has successfully taken up the plasmid. We achieve this by separating the luminescent bacteria from the non-luminescent ones and then giving them time to reproduce by binary fission. Binary fission is a type of asexual reproduction by the separation of the body into two new bodies, where each new organism has a copy of the original DNA. We would then introduce our PET plastic into the culture and leave the bacteria to degrade it. The results were recorded by determining the concentration of CO2 gas produced. We chose this method of measurement as microbial degradation of a long-chain polymers, such as PET, involves the release of CO2 gas. So the concentration of CO2 gas produced is proportional to the amount of plastic broken down.
So these are our theoretical results. We state that the bacterial cultures containing PETs and METs only modified bacteria will break down the least amount of plastic and have the least and have a really low turnover rate. The bacterial culture containing PETs and METs break down the most amount of plastic because the enzymes work in a two-enzyme system.
As with every project, there's also going to be some advantages and disadvantages. So our project aims to use microorganisms to provide a solution to the problem of plastic pollution, save energy, as well as achieve to have cleaner oceans. Today, most of the plastic is incinerated once it has been used, and so useful material and energy is lost. So by the use of bacteria, this energy does not have to be used up, and then materials can still be available.
Unfortunately, there are some disadvantages too. Our modified bacteria is only used for PET because the enzyme that we propose is very specific. We assume that the process is going to be slow as the turnover rate of the enzyme is very low.
So looking into the future, we'd actually want to carry out our experiment and make further discoveries to hopefully reduce the problem of plastic solution. And as stated before, we'd also want to find different solutions for different types of plastic other than PET.
We would also ideally want to modify our enzymes PETase and MHATase to have a higher turnover rate so that they degrade plastics faster and so make the whole process of plastic degradation more efficient.
And finally, we want to apply our experiment here in Luxembourg and measure the plastic content in the Luxembourgish rivers and lakes. And hopefully we can degrade the plastic pollution here in Luxembourg with our experiment. Thank you for listening.
Thank you for listening.
Thank you very much for the presentation. Any questions?
Yes, I have a question. Thank you for your report. How did you estimate the level of degradation that you showed us a few slides ago?
By the concentration of carbon dioxide being produced.
But your work is theoretical, right?
Yes.
And how did you estimate it theoretically? Can you explain, please?
We couldn't do those calculations ourselves. What we only had was theoretical information from research papers, which showed how each enzyme worked.
Okay, I understand. Thank you.
Thank you for your presentation. Can you tell me, do you have any ideas on how to increase the effectiveness of this enzyme and how to influence the rate of the reaction?
I'm sorry, could you repeat your question once again?
Yes. So do you have any ideas on how to increase the effectiveness of this enzyme and how to increase the rate of the reaction?
So as stated before, we said that we would be giving the culture a heat shock. So this basically activates the enzymes to produce a higher turnover rate. So the heat shock corresponds to the turnover rate initially.
Okay, thank you.
Any more questions? Well, thank you then for your presentations.
Thank you very much.
Thank you.
Now we're gonna move to the team from Turkey.
Greetings. My name is Ege Çalışkan.
My name is Murat Işık.
We are from Turkey, and we are here to present our project named Prediction of SARS-CoV-2 Mutations that Increase Contagiousness. The COVID-19 pandemic, which appeared in 2019 and has taken 223 countries under influence, has caused over 2.5 million deaths and over 110 million cases. The COVID-19 pandemic has reached this level of contagiousness because of the quick spreading of advantageous SARS-CoV-2 mutations.
Advantageous variants have occurred especially by mutations composed of a change in a single amino acid. These mutations, just as they can cause change in the structure of the virus, can also affect the efficiency of the interaction between SARS-CoV-2 and ACE2, which is a protein that the virus uses as the first step to enter the human cells.
SARS-CoV-2, which belongs to the subfamily Coronaviridae and the Coronaviridae family of the order Nidovirales, is approximately 150 to 160 nanometers in size, has an enveloped and spherical structure. The positive single-stranded RNA, which carries the genetic material of SARS-CoV-2, is protected in a capsule composed of nucleoproteins. What is the S protein? The red, spike-like structures on top of SARS-CoV-2 are called S proteins. They are alternatively called spike proteins as well. SARS-CoV-2 recognition of the targets on human cells are made possible with the S protein. SARS-CoV-2 starts the infection by connecting to the receptors on the host cells. What are RBD and ACE2, and what do they have to do with the coronavirus? RBD is the receptor binding domain, which is located on the S protein that helps the virus recognize its target, which is the ACE2. ACE2 is a type of membrane cell envelope protein. The strong interaction between the catalytic region of the ACE2 enzyme and the S protein of the SARS-CoV-2 virus is the first step in the formation of the disease. How does SARS-CoV-2 and ACE2 interaction affect the virus mutation? If you are to inspect the visual on the screen, the S protein on the SARS-CoV-2 virus is a Lego piece. This Lego piece connects to the human cells, which is a Lego piece with 2 entrance points. But the antibodies connect to spike in a way that will prevent it to connect to the human cells, just like in the last step. But if the spike protein has mutated, it can connect better to the 2-piece human cell. And now that the protein has 2 pieces, the antibodies cannot connect to the mutated spike protein. Among mutations that occur while the virus is copying itself inside a cell, some grant an advantage. Some of these advantageous mutations allow the S protein to better adapt to the ACE2, increasing the speed of contagion. N501Y and E484K mutations that have been seen since 2020 in England, South Africa, and Brazil allow the S protein on the virus to better bond to the ACE2, and it made some antibodies against the S protein useless. In order to solve this pandemic problem that deeply affects the whole world and causes this presentation to be done online, by feeding the experimental and literature knowledge of biochemical changes caused by the binding and S protein stability data to different machine learning algorithms, we have aimed to detect the SARS-CoV-2 mutations that have an advantage similar to N501Y and E484K mutations. Firstly, we collected the mutation data, then it inspects the SARS-CoV-2 and its target protein ACE2's interactions from scientific literature. Then, we process the amino acid change features in the SARS-CoV-2 mutations. Using the K-means and expectation maximization algorithms in VECA, we aim to predict which SARS-CoV-2 mutations have the same effect as N501Y and E484K mutations. We evaluated the mutations that are grouped together with these 2 mutations in the scope of SARS-CoV-2 and ACE2 interaction.
So, is it even possible to predict SARS-CoV-2 mutations that increase contagiousness? According to the article by Ster et al. That includes 4,221 S protein RBD mutations, around 14% increased S protein to ACE2 binding. N501Y and A484K mutations, which are variants from England, Brazil, and South Africa, are a part of this group. The fact that only 2 mutations have been commonly seen while there are 586 advantageous mutations means not every advantageous mutation has equal chance of being widespread.
In our project, using experimental datasets, we made new datasets with our Python code. These datasets can be found on our GitHub page. I would like to show you our page and explain our datasets. Here you can see the GitHub page we made for this project. Now, our most important dataset that we use to get our final results is this one called Dataset New Features Binding 0 and Up. We have iterated through multiple datasets of our own making and decided this one to be the optimal for analysis. The reason we chose this one is because it has the lowest amount of candidates to select the dangerous mutations from, because we filtered our mutations and selected ones that don't have a negative effect on binding affinity. And it includes the most amount of features to represent each mutation.
The amino acid features we use in this project are polarity, hydropathy, volume, ring number, double bond number, oxygen number, hydrogen number, and molecular mass. The main reason for using this feature is that these features have an important effect on the binding of SARS-CoV-2. In the project, we aim to make a classification based on determining the change in the side chain of amino acids depending on the mutation. VECA is a program widely used in the academic community for data review, analysis, and forecasting. In this project, we used VECA to observe the distribution of data and extract meaning and information from the data.
We introduced our experimental dataset to do different machine learning algorithms. For different group numbers, we investigated which mutations would fall into the same cluster as N501Y and E484K mutations. You can see these algorithms on the screen as well. Now, we want to convey the result of our analysis with these algorithms to you. First of all, we wanted the cluster with harmful mutations to have as few elements as possible, and we achieved this in 10 clusters. For this reason, with the k-means algorithm, we have decided that our data should be divided into 10 separate clusters. E484 and N501Y mutations are in the same cluster, and this cluster has 30 elements. The fact that these mutations are in the same cluster indicates that our clustering approach is reliable. The same way, just like the k-means algorithm, in the expectation maximization algorithm, we wanted to simplify the cluster with potentially dangerous mutations. And we achieved this, just like in k-means algorithm, in 10 clusters. Therefore, with the expectation maximization algorithm, our dataset should be divided into 10 separate clusters. In groupings, we observed that N501Y and E484K mutations went to the same cluster, just like in the k-means algorithm. This indicates that our clustering approach is reliable with both algorithms. As a result of the analysis of the clusters obtained from the k-means and expectation maximization algorithms, it is seen that there are common mutations in the list of the 5 most dangerous mutations. Evaluating the dangerous mutations from both algorithms, we have found these 6 mutations to be the definitive list of mutations with the highest possibility of being dangerous: N501M, N501W, Q414A, N354K, Q498H, and 460K.
In the image on the screen, you can see that the 498 and 500 first positions are on the interaction surface. Among the 6 dangerous mutations, position 501 is repeated. So we emphasize that mutations in this position should be given special attention as the most important result of our project. In our project, where we aim to find the dangerous mutations of the SARS-CoV-2 virus, which causes the COVID-19 pandemic that the whole world is fighting, using machine learning algorithms, using the experimental dataset we obtained from the literature, we introduced the dataset which contains mutations that improve or does not affect binding, contains 8 amino acid properties that we have created with our own Python code to various machine learning algorithms. We determined the k-means and expectation maximization algorithms as the algorithms that give the most consistent results. As a result of our analysis using these 2 algorithms, we found that the optimal value for each algorithm to divide the dataset into is 10 clusters. And we determined the cluster containing the N501Y and A484K mutations together as a set of dangerous mutations. When we examined the cluster of dangerous mutations produced by the 2 algorithms, we saw that there were common mutations in the top 5 most stable mutation lists. When we examined these mutations, we consensusly identified 6 mutations as those with high probability of dangerous effects on the spread of the coronavirus. As the 501st position is repeated in our list and is now one of the most common variants, we emphasize that the mutations in this position should be paid attention to as an important result of our project. The world of science is experiencing a dynamic pandemic process in which any work to be carried out for prediction of the transmission rate and method of the virus will both provide serious benefits and be tested. Developing the possibility of accelerating the transmission of SARS-CoV-2 mutations in this area, especially by prediction algorithms, provides strong evidence that more rich and predictive solutions will be obtained in the fight against the virus. Therefore, such studies will provide guidance regarding the spread and progress of the virus. Here you can see the resources we use in this project, and thanks for listening to us.
Thank you very much for your presentations. Are there any questions?
Thank you for your study. I have one question. Tell us, please, what is the future of your project?
Well, in the future, there are some areas that our project can be improved upon. Let me show you them very quickly. Here you can see we have some suggestions for the future of our project. These suggestions are, by adding new amino acid features, we can increase the number of factors examined and examine their effect on algorithms. We can develop a mutation prediction algorithm. We can use different algorithms and reevaluate the results. The efficiency of the mutations we have found can be tested with advanced molecular simulation programs. In case of encountering the 6 most dangerous mutations we have found, great sensitivity should be shown to prevent the spread of these mutations, and necessary health measures should be taken. These are our suggestions for the future of our project.
Okay, thank you very much.
What do you think? Is it enough data for machine learning and for your program about the virus and mutations?
Yes, we have gathered a lot of data, and we filtered only the ones that we need, so we have a data set that includes only mutations that can improve our algorithms, and it doesn't include any irrelevant data, so it's enough data.
If there are no more questions,
Okay.
Thank you very much. Would you mind please starting your screen presentation? Thank you. And we shall welcome Jorge Paragarcia from Spain.
Hello.
Hello. You can start.
Hello. Thank you. I share the presentation. Yeah, can you see it?
Yes.
Okay, I start with your permission. Hello everyone, my name is Jorge Parra and I study in the Alcántara High School of Alcantarilla, Murcia, Spain, and I'm going to present to you my research project, which is called Portman, a weight problem, the paradigm on soil contamination. Portman is a coastal village of the region of Murcia located at the foot of the Sierra Minera La Unión Cartagena, who is popularly known for its bay, whose environmental problems have been the main concern for the village since the middle of the 20th century until nowadays. In this research, the following objectives were proposed. First of all, to make a research about the historical pollution of the area and to see how it has evolved over the centuries. to analyze soil and water samples looking for heavy metals and to compare the problem and control areas, to determine if soil contamination causes the ecosystem to degrade, to analyze the emblema study and to contrast both sides of the social problem that has emerged directly from the bay, to determine if there is a potential risk for the inhabitants of the Sierra Minera, and last of all, to extrapolate the research to other fields. In order to achieve those objectives, a specific methodology has been used. First of all, the collection and preparation of soil and water samples and their analysis with the technique of the optical emission spectrometry. This took various steps, starting with drying and sieving the samples, so we can apply an acid digestion with 25 milliliters of hydrogen peroxide and nitric acid. So we analyze the micro and macronutrients. Then we'll do some plant cultivation. And last of all, we'll do some interviews to know more about the problem that we are talking about. Two of these interviews was made to the both sides of the social problem. And the last one was made to know more about the historical contamination. But in order to know the context of the village and its relation with minor activities, we have to know its history. Since the Neolithic era, there were small communities here, but it was not until the pre-Roman and the Roman Empire times when Portman had its gold era. But the problem that we are talking about starts in the middle of the 20th century. In 1957, the largest facility of all Europe dedicated to the mineral processing was installed in Portman, and in 1959, they were authorized to dump the residues directly to the bay. So the social protests were rising, and in 1986, a massive protest by Greenpeace went quickly around the world. Finally, in 1990, the dumping activities were closed. With more than 60 million tons and 75 hectares filled up with minor sterile pollutants, we face one of the biggest environmental problems of all Spain and Europe. But which problem are we talking about? We have a series of soils full of minor pollutants, so we face a serious case of soil contamination. But first of all, we should define some concepts. Soil can be defined as the final product of the influence of a series of active factors like climate and topography on a passive and parental material, the bedrock and minerals, which form the superficial layer of the Earth's crust. And a pollutant substance is the one that it's not in its natural environment, or it belongs to the environment, but its concentration is higher than it should be, so it causes negative and counterproductive effects. There is a lot of substances that are pollutant, but in this research, we have focused on the heavy metals, which are elements with a metallic nature whose density range between 4 and 7 grams per cubic centimeter and have potentially contaminating characteristics such as bioaccumulation or biomagnification. In Spain, the normal compositions of the soils is given by the generic reference levels. Now I'll start the experimental design, starting with the soil analysis. There were 14 collection points for soil samples. 7 of them were in the problem area within the mining area of Portman, and the 7 other samples were taken in the control area in the Alcantarilla municipal area. So the preparation took various steps. First of all, we collected and sorted the samples, and then we dried, sieved the samples to take them to the analysis facilities where they spent 2 days in a heater. The problem soils in the vast majority of the samples far exceed the generic reference levels imposed for the area. Highlighting some elements such as arsenic, lead, and zinc, we can see how those concentrations exceed the normal levels that are supposed to have. On the other hand, the control soils are, in general, quite in line with the NGR defined for them. If we highlight the same elements as before, we see that arsenic, as well as lead and zinc, keep their concentration in line with the normal level expected. The following graphics show how in the problem soils, all the heavy metal concentrations greatly exceed the values obtained for the control soils. Besides that, the problem soils keep a great tendency to contamination, as we see in the graphics. So the analysis carried out has allowed us to obtain the concentrations of most of the elements of the periodic table. The final comparison is that all heavy metals increase their concentrations in the problem soils, with the largest increases for arsenic, lead, and zinc. Concerning to the rest of the elements, quite high increases are still observed in the mining area soils. For the water analysis, we took 2 samples were collected in the problem area in the Portman Bay, and the control sample was taken at a tap in my high school. So the only preparation was using a water filter to put the samples directly on the sample carriers. From the results obtained, it's only worth mentioning the high hardness of the water taken in the problem area, as we see in their concentrations of calcium and magnesium. The plant cultivation was made with the idea of analyzing the influence of the soil on the ecosystem and to verify whether or not the presence of contaminants affect the development of life, trying to extrapolate the conclusions to the entire ecosystem. So the idea was to grow crops with soil as the only variable and to add substrate to see the effects. We concluded doing 4 crops of red radish with 4 seeds per crop. The crops started growing on the 12th November 2019, and they were growing until the 6th of January 2020, when the experiment finally ended. Analyzing the leaves, because there was no radish fruit, we can see how those obtained in the problem area are fewer and less heavy. In addition, the problem area crops had a visual appearance much unhealthier than the control ones. So concluding, we can see that the crops developed in the control soils developed way better than the problem ones, and the addition of substrate did not lead to an improvement. So the influence of soil on crops has been clearly shown. The presence of pollutants affect directly to the development of life and the homeostasis of the ecosystems. The main objective on the Emblema study was to compare directly samples from the mining and non-mining area to determine whether or not there is a potential risk for the health of the inhabitants of the Sierra Minera. In children, concentrations are almost the double in the mining area. In mother, quite high increases are still observed, although not as much. In lead, we have a higher concentration in children because they are exposed directly to those residues. Concentrations in mothers can be explained because in the past, the gasoline with lead was allowed. The concentrations of mothers with cadmium can be explained because of the influence of tobacco. And last, for the arsenic, the data would be approximately even, although a small increase could be even seen in the non-mining area. From the interview with the Emblema study technicians, we highlight, among others, the following conclusions. The presence of mining waste is not denied, but many factors should be taken in consideration. So the increases in the mining area are the same as years ago, but with lower values. So for them, there is no potential risk. These ideas clash head-on with the conclusions of the interview with the pump responsible, on which we highlight that the regional administration would have a denialist attitude and would try to divert the attention with impossible action plans. So the solution would be to enforce environmental regulations and keep the residues stabilized. So for him, the population of the Sierra Minera continues to be at risk. In conclusion, the Portman Bay has gone down in history as the scene of one of the most serious contamination cases of all the Mediterranean, where the main pollutants are heavy metals, arsenic, cadmium, and lead, and the main source of non-labor exposure is the digestive one. The Sierra Minera soils, the high cost of the necessary actions have led an unhealthy panorama of pollution, where the Sierra Minera soils fairly exceed the NGR defined for them, and they affect dearly to the development of life and the homeostasis of the ecosystems. Neighborhood protests and social problems have never stopped because they keep finding safety problems with residues. So it could be necessary to think about the consequences of our activities, and a more severe environmental legislation could be needed to solve the problems of the inhabitants of the Sierra Minera. Now I present to you the bibliography and webgraphy used for this project, and I want to thank everybody who has been supporting me and who has helped me doing this project, especially to the Congress organization for letting me presenting the results of my research project. Thank you very much for your attention.
Thank you for your presentations. Are there any questions?
I have a question, if I may. Thank you for your presentation. It was very interesting. What method did you use for quantitative analysis of soil samples for heavy metals?
We used the analysis by optical emission spectrometry. It's called the ICP-OEs. because it's the abbreviation of inductive cold plasma optical emission spectrometry. And we did this on the facilities of the Cebas CSIC Center here in the University of Murcia. And that's the method that we used.
Thank you very much.
Thank you.
May I ask the question? Do you have any suggestions to soil recultivation, for example?
Can you repeat?
What can we do to recultivate the soil, to decontaminate it?
Oh yes, that's a very interesting question that actually we were trying to answer, but the actions that we could do is stabilizing the residues, because it would be impossible to eliminate all the residues because there are more than 65 hectares full of polluting substance. So the bay is completely filled up with minor pollutants. So the main action that we could actually do is to stabilize the residues and try to them to not have an environmental effect directly to the bay. So we regenerate the bay and keep the residues stabilized.
And do you know that in Canada they had the same problem with mining and with industries, metal, for example, industries, and they already recultivate the big area by planting grass and trees and so on. Do you know about it?
Yeah, we were talking with the PAM responsible about this kind of projects, and he told us that we could do a plan for just regenerating some fields of the bay, not the entire bay, but we could do like parts of the bay. We could try to just stabilize the residues and plant some trees or that for the soil to regenerate the natural cycle of the elements of the soil. And we could do this with parts of the bay, and in an ideal case, it would be the final result, it would be all the bay regenerated. But it's true that we have to take in consideration also the economy of the area, because this is a little bit depressed, economically depressed area. So many factors should be taken in consideration. So the ideal case, obviously, is regenerating the bay, but the regional administration would try to do its best.
Thank you.
Thank you very much.
If there are no more questions left, we shall thank our speaker and move to Filip Koljic from Serbia.
Hello, everyone.
Hello, please start your presentation.
Yes. Can you see the presentation?
Yes.
Okay. Good afternoon, everyone. My name is Filip, and I come from Serbia, Department of Chemistry, Petnica Science Center. My research is about novel imidazole-based dyes, their synthesis and investigation of various types of chromism. Imidazole-based dyes are present in our everyday life, and they have many potential applications. And what they have in common is the imidazole functional group. They have various applications, for example, in making photographs, in designing solar cells, for initiating polymer reactions, and they could be used as herbicides, fungicides, and bactericides. What they also have in common is that they usually have various types of chromism, such as solitochromism, thermochromism, halochromism, and thermochromism. Solitochromism is the feature of the substance that it changes color in various solvents, such as shown here in the slide. The change in color is based on the moving of the absorption maximum. If it is moving to the right part of the spectrum, we're talking about beta-chromic shift. And if it is moving to the left part, we're talking about hypsochromic shift. And the quantitative description of solitochromism could be done, for example, by using Kamlet-Taft equation, which is given on the slide, which shows dependence on absorption maximas, on solvent acidity, solvent basicity, and solvent polarity. Halochromism is the feature of substance that it changes color based on the pH value of the solution, and as it is shown on the slide. It can be continuous or discontinuous. What is shown here in the slide is that substance changes its color gradually, and then we are talking about the continuous halochromism, but when the change happens at exactly 1 pH value, we are talking about discontinuous halochromism. And halochromic substances can be used as pH indicators in analytical chemistry. Thermochromism is the feature of the substance to change its color based on the temperature. It can again be continuous and discontinuous, and some examples are shown on the slide. Piezochromism is the feature of the substance when it changes color after applying mechanical pressure. And piezochromic materials have many applications in designing various sensors. And piezochromism, again, can be both continuous or discontinuous. The principal aim of my research was to synthesize methyl and nitro-substituted hexaphenyl B-imidazoles and to investigate their solitochromic, halochromic, thermochromic, and piezochromic behavior. The first step of the synthesis was the condensation between benzyl, substituted benzaldehydes in the presence of ammonium ions. And by that, substituted phenyl imidazoles were obtained. And the second step was oxidative coupling between those substituted triphenyl imidazoles to get the dyes, where the dye with the methyl substituent is called 2A, and with the nitro substituent 2B. Now I will present the results and discuss them. Dye 2A shows solitochromic behavior, as it is shown in the slide, where you can see that its solutions in various solvents are differently colored. And when we look at the UV visible spectrum from 350 to 700 nanometers, we can see that the absorption maximas are in the yellow range, which means that the solutions of the dyes would be purple. And when we are talking about the dye 2B, we can see that its color is also changing from yellow through orange to deep red. And when we look at the spectra, we can see that they have two maximas in the visible part of the spectra. First, in the green part, which is responsible for the red coloration, and one in the purple part, which is responsible for the yellow coloration. Here you can see the results of multiple linear regression based on Kamlet-Taft model, where we can see that the dominant factor for the dye 2A is solvent polarity, while speaking about the solitochromism of dye 2B, we can see that all three factors are equally responsible. And based on the linear regression factor, we can see that solitochromism for the dye 2B is a very complex process. Speaking about the halochromism of these two dyes, you can see the pictures where the middle picture corresponds to a neutral solution, the left pictures correspond to acidic solutions, and the right pictures correspond to basic solutions. We can see that the halochromism of dye 2A is discontinuous. It is yellow in acidic medium and purple in neutral and basic medium, while for the dye 2B, it is yellow in acidic medium, orange in neutral medium, and deep red in basic medium. Speaking about the thermochromism, we can see that both synthesized dyes show thermochromic behavior, where thermochromism of the dye 2A is again discontinuous, while the thermochromism of dye 2B is continuous. The color change in the case of 2A is from yellow to purple, and in the case of 2B, from yellow to red. Thermochromism was investigated only qualitatively, and I can surely say that it happens above 100 degrees. I didn't have enough time to investigate the critical temperature where it appears, but it can be a part of the future research. And speaking about the piezochromism of two dyes, here you can see the pictures before applying mechanical pressure, just after applying mechanical pressure, 2 hours after applying mechanical pressure, and 6 hours after applying mechanical pressure. We can see that the color change in the case of 2A is from colorless to purple, while in the case of 2B, it is from yellow to orange-red. And to conclude, in this research, successfully were synthesized 2 novel B-imidazole-based dyes, and both of them showed solatochromic, halochromic, thermochromic, and piezochromic characteristics, which includes that they have various potential applications, for example, in making photographs, designing solar cells, etc. This research could be extended by investigation of dyes with other substituents than nitro and methyl group, by determining pKa values for synthesized dyes, and hence making them usable as pH indicators in analytical chemistry, and by investigation of photochromism of synthesized dyes. Thank you so much for your attention.
Thank you very much for your presentations.
You're welcome.
The speaker.
So yes, thank you for your presentation. I have one question. What actually in the structure maybe of this substance allows this substance to exhibit such chromism? Maybe it's bond shifts or different interactions or what?
Yes. Here is what happens. We have this B-imidazole derivatives, and when we apply either pressure or temperature or light, there is a homolytic dissociation of this bond, and we get 2 radicals, which then absorb light of different wavelength other than the original molecule, and these are the species that are responsible for the different coloration.
Thank you.
You're welcome.
Thank you for your presentation. Can you please tell us what was the aim of the work? Is it new materials? And what advantages and disadvantages of your materials comparing with existing?
Yes, the principal aim of this research was to synthesize 2 novel imidazole-based dyes, which was successfully done, but also to investigate their various potential applications. There are some known hexaphenyl B-imidazole derivatives, but with other substituents than methyl and nitro groups, and they were not all synthesized in the position 4 of this phenyl group. So this project had 2 aims: to design a new dye and to successfully synthesize them in high yields. And the second one is to combine 2 various steps in a synthetic process, because there were some attempts to synthesize these dyes, but the yields were not that high. So I combined the first step from one previous research project and the second step from the other one, and combine them in order to try to get higher yield. And it was successful because in those researches, yields were around 40%, while in my research, yields were around 70%. Also, the advantage of these dyes are that they can be used in designing various sensors because they have various types of chromisms. So they can be used for both photosensors, pH sensors, thermosensors, etc. But one of the major disadvantages of these compounds is that they were not soluble in many solvents. For example, dye 2A was soluble in only 6 or 7 various solvents, while dye 2B was a little more soluble, but again, the solubility was very limited, but enough for solutions to be deeply colored so their UVB spectra could be recorded.
Thank you. Now it's clear.
Could you say what is the potential for commercialization of your new dyes?
I'm sorry, could you please repeat the question?
Could you assess the commercial potential for commercialization?
Well, these dyes are synthesized from relatively cheap materials, from benzyl, substituted benzaldehydes, and ammonium acetate, which are relatively cheap. The process is pretty fast. I mean, dyes could be synthesized in less than 4 hours in relatively high yields. And so the price is accessible, and they could be used for many purposes. So if somebody produces a dye, they can use them for many different purposes, not only for one. So I believe that these dyes could commercially be available to a larger number of people.
Sorry. Hello, Philip.
Hello.
Can I ask you some questions?
Of course.
What can you say about the toxicity of that products, of that compounds?
In this research, I didn't access that part because I was concentrated on the synthesis part and on chromism investigation. But some of the dyes can be used, as I said in the introduction, as herbicides, fungicides, bactericides, but they are previously synthesized molecules were not toxic for people. Of course, as every other dye, they are pollutants, but with a proper handling, they could not contaminate the environment. So speaking about the previously known dyes, they were not very toxic for people, but for particularly these two, I unfortunately do not have that information because they were synthesized relatively recently, so there was not enough time to investigate that.
Good, thanks. And one more question. What spectral methods did you use to describe your substances?
For recording their UVB spectra, I used the regular spectrophotometer in the visible and near UV range from 350 to 700 nanometers. And for qualitatively describing Kamla-Taft model, I used the linear fit, linear multiple regression fit.
Didn't you do NMR spectra?
Unfortunately, I still do not have the NMR data of these dyes. They were characterized only by infrared spectroscopy and determining their melting point. But as I said, I recently finished this project, and I sent my samples for the NMR analysis, but I still didn't got my spectra. So I'm waiting for them.
Okay, thank you. Thank you.
Thank you so much for the questions.
Are there any more questions? Well, thank you, Philip.
Thank you so much for the interesting questions and discussion.
Yeah, thank you very much for your presentation. Now we are moving to Nikolai Nikolaev Georgiev from Bulgaria.
Me? Hear me?
Hello, yes, we can hear you.
Okay, I'm sharing my screen right now. Can you see the presentation?
Yes.
Okay. I want to, do I have to change it to full screen? Because it will be easier without any technical issues to just change the slides like that. Is it okay?
It would be better if you could do it full screen.
Okay. Can you see it now?
Yes, thank you.
Okay. So I'm Nikolai Georgiev. I'm from Bulgaria. I study in high school in Sofia, and my project is called spermatozoa protamine 1 as a potential biomarker of ram semen quality. So I'll start by identifying the problem. First of all, in sheep farming, it is expensive to raise studs. Those are the male animals used for reproduction. In sheep farming, it is rams. Second of all, the reproductive quality of animals is evaluated based on the quality of their offspring. That means that if we want to evaluate a certain animal for its reproductive qualities, we need to raise it, to have it, to have an offspring by that animal, and then wait until that offspring grows up, and then evaluate it to have some information on the reproductive abilities and qualities of the initial animal. So what that creates is an ineffective use of resources with lots of unnecessary costs, meaning that a lot of times animals are being raised and resources are being invested into animals that turn out to be bad breeders. So what should the solution be? We obviously need ways to drive risk down when selecting studs to be raised for reproduction. And that should be a biomarker that is important for proper reproduction and shows genetic quality. Obviously, that should be a certain gene or gene expression levels because it would both could correlate with important reproductive qualities, and it also means that those qualities can be inherited by the next generation of animals. So we decided to test if the gene expression of a gene called PRM1 could be that biomarker. And next is the question, the logical question, why exactly that gene? That's a gene coding a protein from the protein group of protamines. Those are the main nuclear proteins in sperm, and they play a very important role in the packaging of DNA in the cells in spermatozoa. And why that is important is because the DNA has to be delivered to the egg cell intact, because if there are any sort of damages to the structure of DNA, that would result in a very ineffective, that would result either in a very damaged offspring, or there wouldn't be any offspring at all, because reproduction wouldn't be able to take place. So we hypothesized that the level of gene expression of that particular gene will correlate to sperm quality. And the next question is, what exactly do? We do 3 things. First of all, the variation analysis for the trait, meaning we want to see if that trait varies individually between the different individual animals and age groups, or it is, let's say, a constant factor that is tied to some other trait, for example, age or something like that. And next, we do correlation analysis for PRM1 gene expression levels and sperm motility as well as sperm vitality. Why those 2 indicators? Because they are the 2 most important indicators that we have and use for the evaluation of the quality of sperm and the reproductive qualities. So what is the contribution? That is the first in Bulgaria study of the levels of mRNA transcripts of that gene in sperm of rams from different age groups from the Bulgarian Bulgarian dairy synthetic population sheep. But of course, that could be, with some further research, could be true for all sorts of sheep species. Next, I'll go over the material and methods used. We had experimental samples from 10 rams divided into groups of 1- and 4-year-old rams. In both groups, we had 5 individual animals. And the next step was the isolation of the total RNA from the samples. And then we determined the quantity of RNA in those samples using spectrophotometry. In order to see if we needed to process the samples further before we proceeded doing the reverse transcription and the polymerase chain reaction. Then we obviously did the reverse transcription of RNA into copy DNA, and then we did the real-time PCR. And using the raw results from the PCR method, we used a mathematical method called the delta-delta CT for the relative evaluation of gene expression. And based on the results we had there, we did some statistical processing in order to see how the results reflected the practical correlation and the variation of the trait. And go straight to the results. So the first thing was the variational analysis. We discovered that there were high levels of individual variation of the trait in both age groups, which was very good for our research and for our thesis. And then we looked into the correlation between the levels of expression of that gene and the quality of sperm. In young rams, there was correlation between both the gene expression of PRM1 and sperm motility as well as vitality. And in adult rams, there was the correlation between the gene expression and the sperm motility. However, there was no correlation found between that the levels of gene expression and the sperm vitality. However, that wasn't that important because we were mainly focused on the young rams in order to be able to early evaluate their genetic abilities. So what's the conclusion? We can conclude based on the results that that indicator would be a good biomarker for evaluation of the sperm and reproductive qualities of rams, and what that means in practice and how that could be transmitted in the practical usage in sheep farming is that it allows for the early selection of young rams that would be good studs, and we can dispose of those who are suspected of being poor reproductive quality, so that we can save and cut costs for the high maintenance costs of those animals who could turn out to be bad breeders. And now I'm moving to acknowledgements. First of all, my mentor and supervisor, Elena Kistanova, then the Institute of Biology and Immunology of Reproduction, that allowed me to use the laboratories in order to do my experiments. And then I will finish with the Ministry of Education and Science of Bulgaria and their program ReproBiotech, through which we financed all the materials that we needed to do the experiments in the lab. And that was it. Thank you for your attention. If there are any questions. Are there any questions I can answer?
Are there any questions for Nikolai?
Sorry?
No, I'm asking if there are any questions for you.
Nikolai, do you know any scientific articles about your problem? Maybe not in Bulgaria, but in EU, other countries?
Not in particular for sheep farming. There were, I've read some articles about the usage of that gene in humans in order to evaluate reproductive qualities. However, in humans, there are two main types of that gene, of that protein, and in humans, we mostly have the other type that is not present, that is not so common in rams. The study on humans found no correlation between between reproductive qualities and the gene expressions of that gene, but our research on rams found that it could be in fact used for those animals. Did I answer the question? Okay, thanks.
What future of this research do you see?
So there are a few main things to be done from now on. The first thing is that the research can be done again a little bit later that year, in order to, with the same animals, in order to see how the gene expression levels vary when the same animal grows. So not just between two age groups with different animals, but for example, of the animal when it was 1 year old and when it's 2 years old, to see if that tendency is preserved. And also, there are some other important genes for reproduction in rams, and we could also analyze them in order to paint a fuller picture of different genes that could be analyzed together in order to have a better picture of the reproductive qualities, or a better prospect of the reproductive qualities of that animal.
Thank you.
Could you please tell us what was your contribution in this work, your personal contribution?
Yeah, I did the experiments and the research on different studies that were on the matter of that particular gene in different species, and that was it. I was for the experiments, I didn't do them alone because I was underage at that time and wasn't able to use the laboratory without the help of my mentor. And then we did the statistical processing of the data together.
Thank you.
Are there any more questions? If not, thank you very much, Nikolai.
Thank you too.
Unfortunately, the speaker from France hasn't joined us yet, so we're going to move to Aleš Socha from Czech Republic. Hello.
Hello. Do you hear me?
Yes.
Okay, so, and see the presentation?
Yes.
Super. So, hello, ladies and gentlemen. My name is Aleš Socha. I am from the Czech Republic, and I study at high school Gymnázium a střední odborná škola in the town Frýdek Místek. The name of my project is automation testing of strip silicon detectors for the experiment ATLAS at CERN. This project is related to the planned modernization of a Large Hadron Collider and the ATLAS experiment in 2024. The main goal of this project is to contribute to automation of testing ITK strip silicon detectors that will be installed into the ATLAS experiment during this modernization. In experimental part, I tested ITK detectors with several electronic tests, and I measured the dependence of electrical noise on temperature with high voltage and without voltage. During the testing, I wanted to find the most efficient procedure that can reduce the time of one test cycle as much as possible, but also met all safety standards. The whole experimental part took place in laboratory of the Faculty of Mathematics and Physics of Charles University in Prague. CERN is a shortcut for the European Organization for Nuclear Research. CERN is concerned with the study of elementary particles of matter. Scientists at CERN are studying the elementary particles with Large Hadron Collider, the LHC. One of the experimental halls that is in the LHC is called ATLAS experiment. In 2024, CERN decided to carry out a complete modernization of LHC and the ATLAS experiment. One part of this modernization will be replacement ITK strip silicon detectors. And those were the detectors that I worked with. ITK strip silicon detectors will reconstruct the trajectory of newly formed particles in LHC. These detectors also generate electrical noise, which can distort the measurement values. I also dealt with electrical noise in my project too. In this picture, we can see the old ITK detector, which is now at CERN. And the next picture shows the new ITK that will be installed into the ATLAS experiment. And this is the detector that I worked with. I used the following equipment to test ITK detectors. I used a freezer in which I could set the temperature. Next, I used Julabo, thermometer, air circulator, interlock, and Arduino sensors. In this picture, you can see the empty freezer before the measurements. Next, I put the equipment inside the freezer. And in this picture, we can see the prepared environment, test environment. Now some detail on the test equipment. And the last picture shows the whole laboratory where I worked. I measured the humidity and temperature values with Arduino sensors, which I made myself. I programmed these sensors and I placed them in various places in the test environment. In Observe to be able to control this monitoring system from anywhere, I connected these sensors with Grafana website. On the Grafana website, I was able to control individual quantities in real time. In this picture, we can see the Grafana website. Electronic tests are part of one test cycle. The first test is strobe delay. This test was very important for correct setting of reading electronics. The second test is trip-point gain. The results from trip-point gain test are, among other things, the values of electrical noise on detector. The both electronic tests checked that detector is working properly. In these two graphs, we see the dependence of electrical noise on detector with high voltage and without voltage. The graph on the left side shows the detector with 400 volts high voltage applied, and the graph on the right side shows the detector with no voltage applied. I measured the decreasing dependence on detector with high voltage applied. The magnitude of electrical noise decreased with decreasing temperature. On the other hand, for measurements on detector with no voltage applied, I measured rather random values. And the magnitude of electrical noise was approximately twice as large. When I compare the data from this graph in one table, we can see that the detector with no applied voltage have had approximately the electrical noise twice as large. Now I will introduce you to a more efficient procedure for testing the detectors. It consists of 6 basic steps. I modified the whole procedure during several dozen test cycles. The whole procedure took about 3 weeks. The result is this procedure that reduced the time of one test cycle from original 40 minutes to 20 minutes, and at the same time, it met all safety standards. The main condition was humidity. Humidity shouldn't exceed 80% during testing. Otherwise, the detector would be damaged. It often happened with old procedure. Additionally, you had to wait approximately 4 hours after each test cycle to get humidity drop back to 60%. Thanks to my new procedure, I was able to continue to the next test cycle immediately. I could exchange one detector for another and continue. It saved me about 4 hours of time. In these 2 graphs, we can see the change of humidity over time. The graph on the left side shows the old procedure applied, and we can see that the humidity was 60% at the beginning of the test cycle, then slightly decreased, and at the end of the test cycle, it increased to 90%. It was very dangerous for the detector. But on the other hand, on the graph on the right side, we see the new procedure applied, and we can see that the humidity values was the same at the beginning and at the end of the test cycle. I focused in this project on ITK strip silicon detectors that will be installed into the Atlas experiment during the modernization of LHC. I built a new functional monitoring system that records the humidity and temperature values in real time. The monitoring system based on Arduino sensors, which I made myself. I programmed these sensors and I integrated them to control monitoring script of Holo Laboratory using C++ root program. I also connect these sensors with Grafana website. Thanks to that, I was able to control the monitoring system, the test environment online. Additionally, I tested ITK detectors with two electronic tests. I found I measured the dependence of electrical noise on temperature with high voltage and without voltage. I found the decreasing dependence on detector with high voltage applied. The magnitude of electrical noise decreased with decreasing temperature. On the other hand, on the detector with no voltage applied, I measured the random values. The biggest contribution of this project is a more effective procedure for testing ITK detectors that reduce the time of one test cycle from original 40 minutes to 20 minutes, and at the same time, it met all safety standards. The biggest advantage is that it maintains the same humidity values at the beginning and at the end of the test cycle. Thanks to that, I saved 4 hours of time. These findings can be realistically used in the final testing of ITK strip silicon detectors in Prague. Thank you for your attention.
Thank you very much for your presentation. Are there any questions for Aleš?
Thank you for your presentation. I have one question. I did not understand clearly. What does your detector do?
These detectors will, in the ATLAS experiment, recreate the trajectory of newly formed particles. It means that when in the ATLAS create new particle, these detectors will know where this particle was.
Okay, can you explain the mechanism of this phenomena?
These particles is created from two layer of silicon plate. I don't know if you remember, understand me. The detector is created from two plate of silicon plate, and when the particles goes in across this plate, the detectors know that. Yes, the particles create some signal in the detector, and the electronics reading where is the particle.
Okay, thank you.
If there are no questions left for others, we can move to the next participant. Thank you very much, others.
Thank you too.
Please welcome David Barbin from France.
Hello, I'm David, a grade 12 student at Lycée Douanier-Rousseau. My project was and still is to answer the question, is the study of chemical reactions possible on the scale of a drop? I have been assisted by a few other students and former students who also worked at the scientific workshop. I have also been helped by various partners such as the laboratory Moltech and the engineering school ESIA. I am currently actively supported by Jacques Lang, a CNRS researcher who works with the Laboratoire du Futur in Bordeaux. And last but not least, by the company Fluidgent. Our goal is to replicate a high school chemistry experiment, but in a drop whose volume does not exceed 1 microliter. The first challenge was to find a container suitable for such a small drop. We thought of oil because water mainly constitutes our drops, and it is not miscible with oil. We have shown experimentally with image analysis that with a syringe pump, we can generate in oil perfectly calibrated drops whose volumes can be easily determined thanks to the flow rate indicated by the syringe pump. So we had to find a way to generate drops of reaction mixtures carried in an oil flow once their contents had been homogenized. We imagined, designed, and gradually developed this type of cell, drawing inspiration from research in microfluidics. To make the cells, we use PMMA, an easy-to-machine polymer, and etched 1 millimeter wide channels. We chose the size and length of our channels, as well as the entry and exit points for our fluids. Then, to ensure that the channels were watertight, we covered our PMMA plate with a PDMS plate. It is a polymer widely used in the world of microfluidics because it is very flexible, transparent, and inexpensive. To manufacture the plate, we mixed PDMS with a cross-linking agent. Then, we molded the mixture obtained and degassed it in a vacuum chamber. We finally steamed the mixture to obtain our plate. We then drilled the PDMS plate to insert the fluid inlet tubes. All we had to do then was to cover the PDMS plate with a new PMMA plate to ensure watertightness by screwing the plates together. We now had to ensure a rapid homogenization after the formation of the drops of reagent mixtures. Obviously, we could not use a magnetic stirrer like this one for our small drops. We calculated the Reynolds number for the flow of fluids in our cell. The values were very low, close to 6, which indicates a laminar regime. We checked this with different colored aqueous solutions, and we could see they remained quite distinct. So we should not expect any turbulence in our cells to homogenize the contents of the drops. Then we were once again inspired by what is done in microfluidics, and we engraved channels with bands located just after the point of entry of the drops. In the photo here, the oil pushed by syringe pumps arrives from this inlet. The two different fluids also pushed by syringe pumps arrive in these other inlets. The drops then formed or carried away by the oil. The gradual homogenization of the drops is observed. What happens is that by bending and refolding the species, in each layer diffuse into the neighboring layers. We were then ready to replicate drop-scale high school experiments. We were initially tempted to perform a conductimetric titration, but for that, we needed a conductimetric probe on the scale of a drop. We tried for a long time to achieve it before giving up. The researcher confirmed no conductivity probe of this type is used in microfluidics. So we looked into a new manipulation, the study of a chemical kinetics of which one of the reactants is a colored species. In this case, the decomposition of methylene blue by a colorless solution of ascorbic acid, vitamin C. But to do this, we have to be able to measure the absorbance of our colored drops. The classic calorimeters at our school are not suitable for our drops. So we gradually developed the following device. We pointed the laser at our drops and their image formed on a photodiode, which transformed it into a voltage. An electronic circuit allowed to amplify and then view the electrical signal transmitted by the photodiode. The curves obtained then allowed to calculate the absorbance of the drops. Many problems and questions arose during the gradual implementation of this device. Among them, the following: which light shall we use? First, we had to determine the wavelength the most absorbed by methylene blue. By plotting its absorbance spectrum, we were able to determine that its maximum absorbance was 664 nanometers. To study the absorbance of our future drops, we therefore had to choose a laser with a wavelength close to this value. So we chose a 650 nanometers red laser available in our school. We were also able to verify that our sensor was sensitive to 70% of its maximum at this wavelength. Another problem is precision and stability. We had to aim precisely at the moving drops with our laser. So we attached the cell to a microscope that we had fully adapted. It gave us a good stability, and its stage allowed to move our cell very precisely. Another problem was that we could not place the photodiode just behind the very small drops. The solution was to project the enlarged image of these drops with a camera lens that we recovered from the photodiode placed at the bottom of the box. The black fabric allowed to darken the box and isolate the photodiode from outside light. To test our device, we generated methylene blue concentration drops. After some setbacks, we verified that for not too high concentrations, the absorbance of the drops was indeed proportional to their dye concentration. In accordance with Beer-Lambert's well-known law, our device was reliable. So we generated drops of reaction mixtures. In this photo, we gradually observe the discoloration which reflects the progression of the chemical transformation. The speed of drops is constant. Their position, therefore, gives us the duration since the beginning of the transformation. We have two options to study this chemical kinetics. Either we multiply the devices at different stages of the evolution to analyze the content of the drops, or we try to make a drop oscillate or rotate in front of the same sensor and analysis system. We gave up on circulating a drop in a closed loop in which the drops would circulate for as long as desired. With the encouragement and advice of Jacques Lang, we opted to create a back and forth movement of a few drops in front of our sensor. At this point, we started a whole series of tests which gave us a lot of problems. We tried to oscillate our drops with a peristaltic motor. Many difficulties emerged. We imagined the assembly that would respond to the following simulation. It was validated by Jacques Lang. However, this requires high-performance microfluidic switches like this one that the Fluigen company has lent. We received this material a few days ago. We hope to assemble it soon. The researcher and we are all convinced it will eventually allow us to reach our goal. Thank you very much.
Thank you very much for your presentations. Are there any questions for David?
Have you got any questions?
Could I ask the question? David, can you tell us what was the reason to begin this project? Why the chemical properties in drops?
Because we think that in all the chemical transformations, we use a lot of products that could be very bad for the environment and for the earth. So we thought that we should find a solution to limit all the reactants, for example. And in fact, in the drops, we think it will be a good solution because a drop of 1 microliter is very fewer than a beaker where you have 20 millimeters, for example.
And where can we use this type of reactions? Not only in school experiments or somewhere else?
In fact, there is other utilizations of these products, like in biology, for example, and especially in the inkjet printer, for example, because they need to use the microfluidics to work, in fact, and so there is other utilization that our cell.
David, and have you any prototype for your unit? Any prototype?
I didn't understand your question.
When you design your experiment, have you any prototype, any example, any scientific articles?
In fact, we just can make today we just can make a colorimetric titration. For example, we are trying to make a colorimetric titration, but in fact, when we draw our cells, we just have the help of the ESIA, an engineering school, and so we don't really use scientific work. Just when we make our PDMS plates like this one, in fact we have a process, but we just engrave our PMMA plates in the school and the engineering school.
Yeah.
If there are no more questions left, thank you very much, David. And we are moving to Greta Kaltunova from Slovakia.
Hello.
Hello. Please, David, would you mind? Thank you. Yes, please start your presentation.
No. Hello, my name is Greta Kolcunova. I'm from Slovakia. I attend Gymnasium Leonarda Štekla in Bardejov. My project is comparison of use of the Vidho 2 bed boxes and external heckaries for reintroduction of the brown trout. Questions. Which of the selected method without vibed boxes or external heckaries is more suitable for increasing the population of brown trout in the river Topla and its tributaries? Variables in my project are the use of two methods of planting trout in without vibed boxes and external heckaries. Another variable was water temperature, natural predators, and time, as we do this for several years in a row and find out whether a number of trout is rising or falling. Hypothesis. We assume that external heckaries will be many times better for planting trout eggs in rivers than without vibed boxes. Materials and methods. Without vibed boxes, the patented without vibed boxes is only available trout into the International Federation of Fly Fishers in USA. The without vibed boxes consists of chamber for fish eggs, chamber protecting the alevin allows the hatching alevin to fall into its bottom. This part is designed to be able to digest the yolk sac at the bottom, and when it is available to swim, it can leave these chamber trout the openings in the middle of the wall, leaving the without vibed boxes. The developed fry can escape. In without vibed boxes can help to 500 fish eggs. External heckaries is heckary designed and manufactured by use under domestic conditions. The external heckaries consist of the upper part of the heckary, see which fish eggs, and hatchery which holds for weather flows. The heckary can hold up to 10,000 fish eggs. The eggs are planted on a hatching sieve where they also hatch and digest the yolk sac. On this map is Slovakia, located in the middle of Europe. The project was implemented in this part of Slovakia. It is Kaleť-Bardejov district, and breeding stream Vyška and Lipovec are classified as breeding district of the upper river Topla basin. Brown trout eggs. After fertilization of eggs, they develop eggs in the eye points. How fast the eggs hatch depends, and the weather temperature, cooler weather, means slower development. Fill fish eggs into the heckary and boxes. Experimental procedure, setting eggs. Here you will see photos from the implementation of the project and explanation of how it was implemented. In these photos are the breeding stream Vyška and Lipovec, which we chose to plant eggs. Preparation and distribution of without vibed boxes and external heckaries. The eggs came in oxygen pressurized bags. Filling of without vibed boxes and external heckaries with brown trout eggs. Regular monitoring of without vibed boxes and external heckaries. Hatching alevin with the yolk sac. In this video, we see a fry with yolk sac. It is the C stage of trout when it hatches from the eggs. The fry provides the trout's first nutrition. It contains lipids, carbohydrates, vitamins, and minerals, and that is needed to survive the first days after hatching. Trout absorb the yolk sac to about 2 weeks, when it does not move very much and rests. We can also see big eyes. So the eyes in the eye points are also colored because the eyes are already very visible in this stage. and collecting without biped boxes and external hackeries after hatching eggs. Catch and measurements of annual specimens. Trout is caught by using the electric aggregate. After the analysis, the fish are returned to the water. Stunning by electric current is minimal. After only 5 minutes, the fish are caught again in full activity. Results: we planted 50,000 brown trout eggs in both breeding streams. Black numbers indicate the success of planting eggs in without biped boxes, and red in external hackeries. After 1 year, we had 2,500 in without biped boxes and 7,500 individuals in hackeries. At this stage, there is a great deal of trout because they are very vulnerable. In the second year, we had 1,500 trout from without biped boxes and 6,500 trout from hackeries. We found this data by catch. In the third year, 106,000 individuals are able to reproduce because each female lays 750 eggs. Trout eggs are introduced into 375,000 from without biped boxes and 2,250,000 from hackeries. By comparing these two methods, we found that the hackery is up to 6 times more successful than without biped boxes. Conclusion: with this project, we found that external hackery are more suitable for planting fish in your condition than without biped boxes. We have seen up to 6 more success. Thank you for your attention. And I also thank my parents, my father, without whom this work would be not possible, and Mr. Professor Jan Kleban. Thank you to Amavet, Volnač Tekel Gymnasium, and Slovak Fishermen's Association in Bardejov. Thank you very much.
Thank you very much for your presentation. Are there any questions for Greta?
Hello, Greta, hello. Thank you for your report. It is very interesting. I have a question. Why did you start studying this problem with this fish?
Okay, big problem is big water, nature of predators, and trout in first stage is very vulnerable.
So you wanted to enlarge the population of this fish, or maybe something else?
Yes.
And what is the future of your project? Can you tell please?
Sorry, I don't understand.
What is the future of your project? Will you grow more fish or another types of fish, etc.?
Yes, in the future, maybe salmon eggs and rainbow trout eggs and Timallus maybe.
Okay, thank you.
Thank you very much. Can I ask you some questions? Why do you choose these two methods? And what analog methods do you know?
External hackery is better methods.
Okay, thank you.
Since there are no more questions for Greta, thank you very much.
Thank you too.
We are moving to Muhammad Muratov from Kazakhstan.
Good evening, ladies and gentlemen. Can you see my presentation?
Yes.
Great. Let me introduce myself. My name is Muratov Dinmuhamed. I'm student of Nazarbayev Intellectual School in Aktau, Republic of Kazakhstan, as well as I'm student of Florida Wilton School at the United States of America. First of all, I'd like to congratulate everybody with Cosmonautics Day. 60 years ago was a very important day, not only for Soviet Union, but also for entire humankind. So yes, my team is the alternative methods of growing substrates. The purpose of my research work, is to analyze, research, and develop sustainable and reliable alternative crops growing methods. A hypothesis is if sustainable alternate methods of growing crops are effective compared to traditional, then further research is needed to improve second global problem, hunger. I mean the second global problem established by United Nations. The introduction is the history of agriculture is a section of agriculture that studies the experience of agriculture accumulated by mankind from ancient times to the present. Humans 12,000 years ago began to cultivate plants, producing food in this way. Thousands of years have passed, but the principles remained the same. However, the technologies of traditional cultivation of the crops are more and more approaching their maximum. And as a result, we need to research, develop new reliable methods of growing substrates that are more efficient. So they are, first of all, the hydroponics. The hydroponics is actually not a new principle of growing. The word hydroponics originated in ancient Greece and means walking water, literally meaning, the principle of growing in this way differs from traditional one is in that the substrates receive all the necessary substances and grow in mineral solution without soil. The advantages of this method is actually they're quite big. First of all, it's flexible regulation of plant growth, the simplicity and economy, the fewer words required, because if you create a sustainable hydroponic system, you even don't need to control the. It not depends on the nature. So it can grow for 365 days a year. So then better growth rate and quality of the substrate. Actually, there's some cons of this method. Not all crops are able to grow with this method. It's a need to create a unique, most efficient water solution and installation of equipment indeed. The aeroponics is actually the same method as the hydroponics, but with quite differences. In the Greek, ancient Greek literally mean walking air. Unlike hydroponics, which use water rich in essential minerals and nutrients as substance to support plant growth, aeroponics growing use mineral enriched aerosol to support plant growth and does not use soil. The aerosol is the atomized solution with minerals and that stuff. The advantages and cons of this method is actually same as the hydroponics. So we need to create a unique solution to atomize the solution, and it's called the aerosol. But advantages are also quite big. So this is schemes of the hydroponics basic systems. Just you can do this at your home maybe. This don't need much technologies or skills. Yes. my work and analysis, I became interested in alternative cultivation methods in 2017 when I was in 7th grade. Then I built a homemade hydroponic installation. The results surprised me. Then in 2019, I received permission directly from my school principal for the construction of large experimental hydroponic installation, but project construction was stopped due to outbreak of the COVID-19 disease, unfortunately. This is my notes. You can see that at 7th day, I replaced the nutrient solution caps. In 12th day, we have the increase in the height of the onions and the beans. So you can see my conclusion, that I concluded. I concluded that the hydroponic method is effective both for growing plants from seeds and for plant with modified root. So this is my gallery. First of all, this is my experimental homemade hydroponic system. You can see the crops going out of the, the system. This is the kind of installation process of the huge experimental hydroponic system at my school. Here the process, here the construction. And in conclusion, I can say that the alternative methods of growing crops is very reliable for us. Yes, because only at this point, there are nearly 7.8 billion people, and this number is multiplying in geometrical sequence. All of us need food to live, obviously, and our children and grandchildren need this also. Our goal is to reduce hunger by developing these sustainable methods. By accurate compound for all substrates is required. Mistakes may cause critical consequences. This is some citations. Thanks for listening. Yeah, now the questions.
Thank you for your presentation. I have one question. Please tell me what nutrients did you introduce to your system?
I used the onions and beans. You mean nutrients? Yes. My solution as a nutrient consists of water and minerals such as potassium, calcium, the several minerals used to primarily used to grow, need plants to grow.
And what source of nitrogen?
Source of nitrogen was the compound of nitrogens. Yeah.
What form of nitrogen can be accumulated by plants?
Actually, I used the potassium nitrate to it's a mineral source in my solution. From the potassium nitrate, the plants, the substrates can use the the nitrogen from the solution.
Thank you.
Of course, with the different proportions that calculated by me to each of the plant. For the onions and beans, there's different solutions.
Okay, thank you very much.
No problem.
I have one more question. Please, can you give us a comparison of growth speed using your methods and the usual method?
Actually, unfortunately, these observations are not consistent in my work because, as I said, I was in process of installation my huge experimental system, but because of the COVID-19 outbreak, it's impossible to do that now especially in my school. So I'm really sorry because of lack of some information, but I will do this as soon as is possible.
Okay, thank you.
Are this all of the questions?
And can we use this method of crop production by the in the large scale?
Of course, indeed. There is the main, actually, this main goal of my project to calculate, to know the advantages of this method, to use it at the large scales of the production, to produce a large amount of food to sustain our nation and sustain the humankind at all in general.
But to use this method, you need special greenhouses, for example.
Yes,
you need to build roofs. You need to build special channels for water. Is it possible for large scale? For example, for wheat production.
For wheat, yeah, yes. Actually, the hydroponic system is possibly the best way to grow wheat in the large scales. But as I said, in the concept of the methods, it needs installation of the actually we need a greenhouse, the lights, the thermometer, the regulator of temperature, and etc. But yes, it is very possible to do this at large scales using the large equipment.
Concerning this question, did you maybe visit some fabrics that use hydroponics or aeroponics method in large scale?
Yes, actually my mom's friend have the hydroponics farm in my city at Aktau. So I visited this farm, and this is why I suppose that it's very possible to do this at large scales, to produce a large amount of food very quickly and effectively.
How much, what is the profit of your mom's friend's factory?
That's a quite interesting question, because for the first years of the working, there is actually no profit, because you need to develop a good quality solution to the best efficiency of growing rate. But when you did the researches, when you create the solution, when you create the atmosphere to growing, this is a quite big profits, but unfortunately I don't know the numbers. But as my mom's friend says, it have a quite big profit.
So, for example, he can maybe sell his products to supermarkets.
Of course.
Is it enough to go to supermarket with your own grown crops?
Yes, all the materials, all the food, crops, and etc. Moved into sell to the local supermarkets, but it can actually be exported to another countries as the original method, the usual method of the growing crops. all this applicable to the alternative method like hydroponics and aeroponics systems, all of this.
Thank you for your talk.
Thank you for questions. Any more questions?
Thank you very much for your presentations, for your answers. Now we are moving to Alina Pleskevich from Russia.
Thank you for listening.
Hello.
Hello.
Do you see my presentation?
Yes, clearly.
Okay. Dear members of the jury and all present, let me offer you the research work about the localization of amongst larvae, Lampetra planary, in the Tega River in connection with habitat conditions, new data for the monitoring of the Red Book. European brook lamprey listed in the Red Book of Kostroma region as a rare species, Red Book of neighboring areas, and the second annex of the Berne Convention. Lampetra planary is the sole representative of lamprey class inhabiting in Kostroma region. It is a small, so sensitive and demanding on the purity of water species that make it one of the most effective instruments of bioindication. Abundance and distribution data of European brook lamprey in Kostroma region are extremely inadequate, and the information of special localization in the scientific literature is practically absent. In this case, this research work is important both for monitoring of Red Book species and for planning specially protected natural areas. The goal of our works was identification of patterns of localization of European brook lamprey larvae in connection with ground, spatial distribution, density of benthic organisms, and dead organic matter on the estuary section of the channel of the Tega River. Objectives you can see on the slide. Sampling was carried out in the summer in 2018. On the estuary section of the channel of the Tega River, the length of about 600 meters. Sampling stations, a quantity of 12 hydrometric gauges were evenly spaced in 50 meters from one to another. Culling of benthic organisms was carried out with standard fishing gear, water net and bottom scraper, which let us evaluate the abundance and the composition of its inhabitants of the reservoir, and it is quite consistent for goals of field studies. All material were put to the plastic tray, was sampled with the tweezers, and transferred to glass sealed jars, which were marked. Unknown instances were fixed by solution of alcohol glycerin mixture to definition in the laboratory. After counting, larvae of lamprey were put back into the river. Offsite data processing and analysis you can see on the slide. According to the modern taxonomy, European brook lamprey relates to the family of lampreys, order of lampreys, class of lampreys, superclass of cyclostomes. Larvae of lamprey prefer sealed sections of the river where they lead a hidden lifestyle, burrowing into the ground. Lampreys eat small benthic organisms and detrital deposits. Their entire life cycle takes place in the river. European brook lamprey is a non-parasitic species. It has no commercial value. In Kostroma region, we can see European brook lamprey in the rivers Tsekha-Nelka-Nesha in Kalagriv, Kostroma in Buy, Poksha in Sudislavl, Shacha in the Kostroma region, and after our research work in the river Tega in Nerechta.
During the study, 101,032 benthic organisms were caught or accounted. They relate to 45 species from 5 taxonomic classes. The dominant class is insects, which consists 5 orders. You see that Brooks order has the biggest species richness. After calculations, the index of dispersion, sharply aggregated type of spatial distribution was identified, both for total population and species richness. Species prefer inhabiting in coastal sections of the riverbed, which are rich for silt or detrital deposits, characterized by a low water flow rate, as a result, better water prehitability. Sharply aggregated type of spatial distribution for European brook lamprey was identified too. Lampreys were seen in 13 sampling stations. The total number of lampreys was 27 individuals. European brook lamprey in the larval stage inhabits in coastal sections of the riverbed, which are rich for silt and detrital deposits, characterized by a low water flow rate. As a result, better water prehitability. The ground is important for lamprey's larvae. They don't live in the sand without silt or detrital deposits, in stony sand and pebble ground. The relationship between localization of European brook lamprey with density of benthic organisms and dead organic matter manifests with different strengths of correlation. As we see from the graphs, both interconnections have a positive trend, but the correlation with density of benthic organisms characterized like moderate, whereas with the TBE index characterizes like low. You can see the conclusions which were made earlier in the course of the work on the slide. Thank you for your attention. I am ready to answer for your questions.
Arina, thank you for your presentation. I have some questions. You showed us several cartograms, but can you explain please how did you, where did you get them, or if you did them by yourself, how did you count the number of lampreys on your cartogram?
All cartograms we did ourselves. We did it with special programs like Passage 2 and Statistica number 10.
Oh, I mean, how did you count the number of lampreys? Where did you get the number of these fishes?
How did we get the number of the species?
Yes.
We count them from the river. We went to the river, then trying to we use their fishing gear. We use their water net to find their benthic organisms.
And were you sure that you found lampreys and not another type of fish?
Sorry, I haven't caught your question.
When you were on a river, you were looking for fishes, right?
No.
No?
No. I study benthic organisms, insects or different worms which are living in the river, in the bottom of the river.
I see. How do they correlate with lamprey number?
how did we get 1,000, more than 1,000 species? We have caught them.
Okay. Okay, thank you.
Thanks.
And how can the results of your research help to protect lampreys?
I consider that our results can help for planning of protected natural areas. So our results have already used in the new edition of the Red Book of Kostroma region. So if we see that population is growing, or it will be great. So we need the trend of positive growth of population.
So you will continue your research?
Of course.
And what is the area of lampreys habitation? Is it unique for our region, for European region?
I can say that European brook lamprey live only in the rivers. So this species isn't unique for our region, but it is really rare. So we need to protect them.
Any more questions for Arina?
Just one more question, may I ask? Do you know are there any more groups that do the same as you in your region?
In our region, we don't have a lot of scientists to study the rivers. Almost all small rivers in our region are unknown. In this case, I think that it is important to protect and to learn something about the benthic organisms, because they are bioindicators.
Okay, thank you for your questions or answers. Sorry, answers and your report.
Thanks for you.
Any more questions for Arina? Well, thank you, Arina, very much for your presentation.
Thanks.
All participants had their presentations today. So Arina was the last one. Next session will be on April 14th at 1:30 p.m. Thank you very much. Thank you everyone for participating in this meeting.
Thank you.
Thank you. Bye.
Goodbye.
See you later.